Hello students, today we are going to learn a new concept, that is DNA fingerprinting or DNA profiling, which is coming under your chapter 6, molecular basis of inheritance. What is DNA fingerprinting? DNA fingerprinting is a technique of determining the nucleotide sequence of certain area of DNA which are unique to each individual. So DNA fingerprinting is a technique of determining nucleotide sequence of certain area of DNA which are unique to each individual. Okay, hence each individual has unique DNA fingerprinting. Okay, DNA fingerprinting is same for each cell, tissue, organ of an organism. It cannot be altered by any known treatment. Okay, Alex Jeffrey in 1984 invented DNA fingerprinting technique at Leicester University. UK. Got it? Then come to principle of DNA fingerprinting. Principle of DNA .1% of total nucleotide gives the individuality to each individual. That means only 3 million base pair are responsible for individuality. Okay, in human genome possesses numerous non-coding, numerous small non-coding but inheritable human genome possesses small, numerous, non-coding but inheritable nucleotide sequence which repeated many times. These are present near telomere, centromere, near Y chromosome, and heterochromatic region. Got it? The area with same base same sequence of base pair, the area with same sequence of bases repeated several times. The area with same sequence of bases repeated several times are called repetitive DNA. Repetitive DNA or satellite DNA or satellite DNA. Repetitive DNA or satellite DNA. According to tandemly repetitive units, satellite DNA are two types. Satellite DNA are two types. One is mini satellite, another one is micro satellite. According to number of tandemly repetitive units. In mini satellite, this number is varies from 100 to 1000 times, whereas in micro satellite this number is varies from 10 to 100 times. Got it? Satellite DNA shows polymorphism. What is polymorphism? Remember, the term polymorphism is used when a variant at a locus present with a frequency of more than 0.01 population. The term, remember, the term polymorphism is used when a variant at a locus present the frequency is more than the population frequency is more than 0.01. Got it? Then come to the satellite DNA or the non-coding DNA or mutation in non-coding DNA have no any immediate impact, whereas this mutation in non-coding DNA have piled up and form the basis of polymorphism, basis of DNA polymorphism. Got it? Short nucleotide repeat This SNR are very specific to each individual and the number is vary from individual to individual. It is also called as VNTR, variable number tandem repeats. Also called as mini satellite. The VNTRs are inherited from parents and act as genetic marker in personal identity test. Half of the variant are coming from the mother and another half are coming from father. Got it? Then come to technique of DNA fingerprinting. Technique of DNA fingerprinting. First of all, DNA is extracted. First of all, DNA is extracted from nuclei of cells, maybe blood cell, spermatozoa or hair follicle cell. Then this DNA are cut into several fragments using restriction endonuclease. This fragment may contain behaviors. Okay. Then DNA cut into several fragments using restriction endonuclease. Molecular scissor. This fragment may contain behaviors. Got it? Then these fragments are separated according to their size by electrophoresis. Fragments of DNA are separated according to their size by gel electrophoresis. Got it? Then fragment with a particular size. Fragment with a particular size multiply by PCR. Then these double-stranded DNA fragments are treated with alkaline chemicals. It converts double-stranded DNA fragment to single-stranded DNA fragment. Got it? Then double-stranded DNA treated with alkaline chemical. With alkaline chemical which convert it into single stranded DNA. Okay. Then this single stranded DNA also contain the variants. This single stranded DNA then transfer onto a nylon membrane. This single stranded DNA fragment transfer onto a nylon membrane. Then radioactive DNA probe. Radioactive DNA probe. Radioactive DNA probe having repeated sequence complemented to VMTs that present in the single stranded DNA fragment complemented to VMTs are poured over the nylon membrane. Are poured over the nylon membrane. Then this radioactive DNA probe binds with the VNTers. Okay. Then radioactive DNA probes bind with single stranded DNA fragment having VNTs. Okay, then excess radioactive 4 are washed. Excess radioactive probe are washed. This technique is also called as southern blotting. The hybridization of radioactive probe with a single stranded DNA fragment having VNTRs are called southern blotting. Got it? Excess radioactive probe then are washed. Then the radioactive probe the nylon membrane. Then the nylon membrane exposed to X-ray film. Then the nylon membrane exposed to X-ray film. Okay. Then the portion. Remember the portion where radioactive probe binds with single stranded DNA fragment are exposed or developed as dark band. Then the portion on nylon membrane where radioactive probe hybridized with VNTR are marked as dark band. It is called as autoradiography. Okay, this dark band represent a DNA fingerprinting or DNA profiling. Got it? Then come to application of DNA fingerprinting. Come to application of DNA fingerprinting. Application of DNA fingerprinting. One person is distinguished from another using this DNA fingerprinting. This is known as individuality test. Identical twins cannot be distinguished using DNA fingerprints as their genetic makeups are same. Got it? Then paternity and maternity dispute. Paternity the real genetic or real biological father and the real biological mother are identified using this DNA fingerprinting. Then hereditary disease. More than 1200 diseases are identified using DNA fingerprinting. Then forensic. Detection of crime and the legal pursuit are done using this DNA fingerprinting. Then in sociology, identification of racial group, their origin and their history of migration are studied using this DNA fingerprinting.